Hello, and welcome to the second lecture on microbial nutrition, where we get to see how microbes grow. All right, so this is where I explain, um, following up from our previous lectures on that how bacteria grow through binary fission, what's actually involved in that, what it looks like when you multiply it out over some generations. And then we get into biofilms. I kind of explained biofilms in the intro, but you'll actually get to see the picture on how they form, how they spread, and a little bit more information of that spelled out. Let's head into it. So we've mentioned binary fission before when we mentioned prokaryotic cell division because we went over the cell and the cell features there. So we wanted to learn about it then and how a cell grows and divides. And so we're going to hit it on it here again because this is in the nutrition area and the metabolism area. So basically, if you give a mouse a cookie, what will happen? If you give the bacteria food, this is what's going to happen. It will replicate its DNA, form a septum, separate, and divide into two daughter cells. The science is a little bit more complicated than saying it separates. All right, we'll learn in our genetic chapter that the DNA is replicated through a rolling circle replication. Okay, and so that's how we get the two sets of DNA up there. Remember, we have to replicate our DNA first. That's the first step. But as we start to form two daughter cells, we need additional help. We get these proteins, FTSZ proteins. They come, form a septum, allow it to divide, and then they go back to what they're doing. Okay. The name FTSZ protein is not going to be important to remember. Okay. What's going to be important to remember is that, again, it is protein that is doing it. You need to have the protein go through the central dogma, DNA to RNA to protein, and that protein is one of our essential biomolecules, okay, because they are the ones doing the work, okay? No protein, no cells, okay? So remember this because it's more how they divide, yes, but there's a lot of factors that we've already learned that go into this slide, okay? And protein, peptide bond, ribosomes, all kind of highlight here what we need to know. All right, so as bacteria grow and as they split through the protein action, they one cell becomes two cells become four, and four cells become eight. Okay, and it all happens rather quickly. Okay, as you can see here above me, E. coli is only 20 minutes. And as I mentioned in the lab, that 20 minutes is affected by an agitation action, by being at 37 degrees Celsius, by giving it the proper nutrients that it needs, etc., etc., etc. Okay, any deviation from that, you will not get a complete 20-minute generation time. Okay, however, given the optimal conditions, okay, E. coli will be 20 minutes. Okay, given the optimal conditions, Mycobacterium tuberculosis gets 15 to 20 hours. Okay, so if you're going to grow even 3 cells divide 3 times, that's going to be nearly 3 days. Okay, whereas leprosy, the disease that causes leprosy, is 14 days. Okay, so we have a long difference of window in generation time. So be lucky that we use the bacteria that are closer to 20 minutes in microbiology lab than are 14 days. Okay, but the matter of fact here again is that this is only after 3 rounds, and we're already at 8. Next time, we're at 16, 32, 64, 128, 256. You get the idea. All right, so it goes pretty quick. And that is why bacteria grow overnight. Okay, and this is very important. Okay, because this exponential growth explains the log phase over there. Okay, number 2, our log phase is directly related to how fast they can grow. From 2 becomes 4, 4 becomes 8. All right, so that's how we get the log phase. Now let's kind of explain these other phases. Okay, so we just explained if we're here during our log phase, 2 becomes 4, 4 becomes 8. We're growing really, really fast. But what's happening here? Okay, there's really just no increase in living cells happening there. Why? It's getting acclimated, it's acquiring nutrients, the cells are growing before they actually replicate their DNA. They need to become happy. Once they're happy, they replicate their DNA. And if we think about that, how narrow this graph is, that it already reaches stationary phase in just that short x-axis width, that lag phase is also pretty short. Okay, so the lag phase doesn't take too long, but eventually the cells will enter the log phase and start to divide. Okay, so what's happening in stationary phase 3? Well, if I look at it, it's staying straight. Let's say one cell divides, and if that happens, I would keep going in my log phase, right? So one cell becomes 2, 2 becomes 4, I'm still in my log phase. So what's happening in our stationary phase? Well, if I made 2, then this one's going to die just as fast as I made 2. Okay, or let's say I became 2, then these 2 over here die. Okay, some way, somehow, the net number of growth equals the net number of death, right? So that's how you can explain that in your head mathematically. So, and it says it right here too, and that's the stationary phase. Okay, now this graph is very important. There will be multiple questions on the final exam about bacterial growth curve. That's why we learned it before in the cell chapter, and we're learning it now. There's going to be multiple questions on it. And if you are confused at all, we can go over it together again. We can go over different scenarios of what happens if you give some of these thermophiles colder temperatures, what happens if you give them warmer temperatures, and then identify their growth on it. Okay, so really kind of understand what this graph is meaning and why we're presenting it before we do hypothermophiles and thermophiles like you see around me. Okay, then we get into the death phase and decline phase. Okay, so just as we explained number 2, our log phase, we're going to get into our death phase. Okay, and so instead of 1 becoming 2 really quickly, 1 becomes 0 rather quickly. Okay, and so the number of new growths is outpaced by the deaths. Okay, and again, you see exponential decrease. Okay, so it's an exponential decrease again. Okay, and how do I know it's all exponential? Again, this is a log scale. Okay, always got to look at your axes. Okay, if it's a log scale and you see something going up and down, that means it's going up and down really, really fast. Okay, remember 3 pH is 1,000 times more hydrogen ions, right? So This is the same kind of thing. It's going really quick. Death phase. All right, switch in to biofilms. Okay, very, very closely related to that growth curve. Okay, as those cells are growing and dividing, they are encountering other bacteria in the environment. Okay, unless it is on a TSA plate in the hands of a microbiologist, most of the time it's going to encounter another bacteria, another microbe of some sort. Okay, so it needs to prepare itself for that encounter, and it doesn't want to die, in a sense. Like, it wants to keep growing and dividing, just like humans. Okay, so we are preparing for a reversible attachment of cells, and that happens in seconds, right? Seconds. So right away, they're on a surface. They say, I'm going to stay here, attaching. Okay, just like viruses, you need to attach. Okay, attachment is so important, which is why soap works. Okay, because soap just is going to loosen that attachment and let those things slide right off. Okay, it doesn't kill anything, it just makes them go away. They can't attach. Then they become irreversibly attached. Again, that can happen within a second or minutes. All right, then they grow and divide. All right, so again, this would be the lag phase. And then as they enter the lag phase, they grow and divide. All right, and as they're in this phase, they even realize they're going to run out of nutrition. So they start to form secondary colonizers to disperse. Okay, and the whole thing about it is that they make EPS. Okay, which forms water channels. It helps form these biofilms. Okay, and it's just another way for the cells to be protected against desiccation, antibiotics, all that kinds of stuff. But note all the different types of bacteria that are in the biofilm here. Okay, many, many different kinds, not just one. Okay, but within a matter of days, we're already spreading to another location, maybe a foot away, maybe 2 inches away, but that's kind of the idea behind biofilms. Okay, and again, you want to associate this with catheters. Okay, the catheters are where you will find biofilms. Okay, and those are HAIs. Okay, or hospital acquired infections or nosocomial infections. Okay, I mentioned quorum sensing in a little bit. And quorum sensing is basically, a quorum is like a certain number of people at a meeting in order to hold the meeting. If you have a party of 20 and only 2 people show up, you don't have a quorum, you postpone the meeting. Okay. Same kind of thing with biofilms. If you don't have enough other surrounding bacteria, you won't have enough of a quorum to make a biofilm. Okay, so quorums and biofilms and catheters. Okay, those kind of all go together.